Okay, I'm recording now. I forgot to hit record. So now I'm recording, and I'm going to go back into sharing my screen. All right, so effective studying. I'm going to spend a little bit every lecture talking about that because little tips and tricks that you can try, because I think if it can help any of you in the class, then that's a good thing. All right, so and have fun with your studying. I make a lot of jokes when I'm studying. I link it to laughter and silly little things, and that really helps me remember things. It's amazing how if you get silly with what you're trying to learn, it really helps. I had students a couple semesters ago in Plant Phys, when I get into the light reactions, it's a very precise step-by-step process that you have to learn, and they made it into this big story, this big silly story. They studied as a group, and they started like creating this story around the light reactions, and they took the test, and then weeks later I asked them to repeat the story back to me, and they knew it cold. I mean, they knew it absolutely, and they did well on the test. They got an A on that question, and then weeks later they still remembered it, and they were laughing about it. So I was so pleased to see how well that worked for them. Okay, so now we're going to get into Plant Architecture 101. We're going to learn a little bit about the plant's basic anatomy to orient you so that when we start getting into the physiology, the plant body will make more sense to you, and you'll be able to imagine what's going on a little bit better. Because obviously a lot of the plant physiology we talk about, it's all internal to the plant. So we need to go inside. So what we're looking at here, on your end, can I just ask, like, is the Zoom blocking any of my PowerPoints? Does it? Okay, you can see it. It's like
no, we just see like the people on the side, and then your PowerPoint is most of the screen.
And can you move the people out of your way at all, or is that all on me? I forget.
Yeah, you can put it in speaker view too, so we just see you in a little box in the corner.
Oh yeah, that's right. I do that when I'm at Zoom meetings. Okay, I'm used to Zoom meetings, but it's been a while since I've done Zoom lectures, so I'm just trying to refresh my memory. Thank you for the feedback.
You're all good.
Okay, basic plant anatomy is being shown here, and this, for those of you who have taken Plant Kingdom, this is going to be old hat, but you've had a whole summer off, so it's good to hear it again. So we look at plants and we tend to divide them by what's happening above ground versus what's happening below ground, because those are super different environments, obviously, and very different types of physiology happen in certain circumstances. Very different things can be happening in the shoot area, the stem and leaves, versus what's happening in the roots. And so the idea here is that the plant body has certain areas of its body that has pretty active cell division and growth. And a couple of the hot spots are the shoot apex, all right, the shoot apex. So you have very active growth. There's a cluster of cells here that are almost constantly dividing and growing and adding length to the stem. And then you have a root apex, again, a cluster of cells that are very precious because their job is to divide, and increase the length of the roots. Okay, we also have areas, depending on the plant, you know, if you have had the great fortune to go hiking among the giant redwoods or the sequoias last summer, was it 2 summers ago? 2 summers ago, I went to Yosemite National Park after a conference and saw the giant sequoias, and it was definitely one of the top experiences in my life. It was awe-inspiring. And you look at the girth on those trees, and you recognize that there has to be some way for the plant to grow out in girth and diameter. And so we do see that there is precious cells, rings of cells in the stem tissue that will allow for some plant species to really increase the diameter of their stem tissue. But not all plants have that. If you think about, you know, some of the herbaceous plants that don't have, you know, bark and wood, they don't really have that. And so it just depends on the plant you're thinking of, all right? But the big picture idea is, in addition to the shoot apex and the root apex, there is a way for the stem, there's active cell division going on to allow for stem diameter growth. And then this term here, axillary bud, I think you can see my, actually let me get my laser pointer. the axillary bud, the red laser pointer, hopefully you can see. So they're pointing to this little bit right here in the crux of the stem versus this leaf that's coming out. So that is what we call a node region, right where you see a leaf or a branch coming off the main stem. That's called the node region, where you will find an axillary bud. This is also can be very active cell growth. Right now it's quiet because it's just little and tiny and just kind of sitting there and dormant. But if someone came along and started like cutting off these leaves and cutting off the shoot apex, then this axillary bud would go, oh boy, you know, we just got attacked or eaten by a rabbit or whatever. I need to grow more tissue. And so this axillary bud will come out of dormancy and start growing. And so there's different areas of the plant body where you see active cell division happening, and then certain areas might be dormant for a while, but they can ramp up and become very active. All right. Then let's see, the internode region is just that part of the stem that is in between two node regions. So inter means between nodes, you know, inter, between. So that's no big deal. But and then they've peeled apart the plant here, and they're peering inside, right where the vascular tissue they're showing when you look internally. We're going to get way into vascular tissue in a little bit. So that you're going to learn a whole lot about. The vascular tissue is like our blood vessel system. It allows for transport of water and minerals through the whole plant body. Obviously really, really important for any multicellular organism that they have a way to transport nutrients and chemical signals around the whole plant body, including plants. They have to do that too. So we'll get way into vascular tissue. And then depending on the plant, there's different designs to the root system. This one is showing a very prominent taproot. So there's one big large root, the taproot, and then smaller lateral roots coming off of it. But this is not the only design. There are all kinds of designs of root systems. Some roots don't even have a taproot. It's just all roots that look pretty much all the same size. So often they draw it this way, and it's fine, but I want you to recognize that root systems have a tremendous diversity in their morphology and their overall design. And so we can't always think about it this way. All right, what else? I think that's it on this. Oh, root hairs, really, really important. Last night I was in the greenhouse. I was repotting my plants, my poor office plants. They suffered all summer long because I couldn't get in there very much. So I decided to give them a little TLC. But one thing I do recognize is that when you transplant a plant into a bigger pot, you always damage the roots, and especially the root hairs. You'd end up stripping them off a lot. And so when you transplant your plants into a new pot for your dorm or home, remember to water it consistently for several days. Give it little drinks of water so the soil stays moist without being, you know, soggy, because you have to give the plant time to regrow the root hairs. The root hairs are the real workhorses when it comes to when the plant has to suck up water from the soil. It's really the root hairs that are doing that. And when you strip all those off, the plant has a much harder time efficiently taking up water. So they need nice moist soil until they can regrow those hairs. Okay, let's zoom in a little. So plants have a cell wall around their cells. We do not, all right? We just have a cell membrane. But we have an endoskeleton, so that's great, right? We have a way to fight gravity. We have a way to have a nice support system through our body to give us structure. Plants don't have an endoskeleton. They don't have an exoskeleton like insects do. But they have all these cell walls that are around the plant cells. And there's different kinds of cell walls, and we'll get into that. So really, this is their structural support. They don't have a skeletal system. So this is the way that they have designed for structural support. So there's essentially two types of cell walls. There's the so-called primary cell wall. That's the one that the plant is born with. It's pre-programmed. When you look at a seed, and you imagine all the cells that make up that seed, primary cell walls, okay? When that seed starts to germinate and grow, every cell is gonna have a primary cell wall. Primary cell walls, when you look at them under the microscope, they're thin in diameter, and they are always surrounding cells that are growing, they're active, you know, all the young cells that start sprouting from a seed, they're primary cell walls that are surrounding those cells, okay? Then, some cells in the plant body will have what we call a secondary cell wall, all right? So all cells have a primary cell wall, and then some cells have a secondary cell wall. Secondary cell walls are thicker in diameter and they are stronger. They're components that make up the cell wall. There's a little extra oomph in terms of what kind of chemicals are in there to really add some extra structural support, all right? And we'll look at this in a minute under in some subsequent slides, all right? So if you have cells that are destined to have a secondary cell wall, that secondary cell wall doesn't get laid down and formed yet until the cell is mature and it's to its full size. And it's like, okay, I've fully grown, I'm at my size I'm destined to be at. Now I can start laying down a secondary cell wall, all right? And when, well, we'll get into that in a minute. And then when you think about, okay, there's all these cells, right, that are growing and forming the plant body, you have to have a way for them to stick with each other and form tissues. And so the middle lamella is really the section, when you look at plant tissue, the middle lamella is the area between two cells that's made up of special chemicals that pretty much, if you want to think of it as allowing the cells to kind of cement to each other and gel with each other, so to speak, so that you end up with a coherent plant tissue. And we'll look at this as well a little bit, okay? What happened to my, oh, there you are, okay. And so I like to think of it as a way to cement all the neighboring cells together to form a nice tissue in that area of the plant body. And pectin is the major chemical component of the middle lamella. And if any of you have picked strawberries or raspberries or blueberries and then you make jam and jellies, you probably added pectin to that. And that's what you were doing. You were adding plant pectin that they harvested from plants. And for the plant, they use it to create this nice tissue of cells. When we use it, it's to thicken up and kind of gel up the jam and jelly. So you can thank plants if you've had some nice jam or jelly in the summer. All right, so here is a mock-up sketch of 3 neighboring plant cells. And you can see the cell membrane is thin and of course very dynamic. And then you have, I tend to say cell membrane, by the way. A lot of textbooks say plasma membrane. I think that's a newer term. It just never really sticks with me. I tend to say cell membrane, but plasma means the same thing. Then you can see the primary cell wall is the outermost cell wall, all right? You can see it's thinner. So that's what, you know, the plant is born with, so to speak. They all have a primary cell wall. And then some cells make the decision genetically, oh, okay, it's my turn now to put down a secondary cell wall. And you can see that's actually laid down to the interior of the primary cell wall. I remember when I first learned that, I did not know that. I always thought everything would go to the outside, but it's made into the interior behind the primary cell wall, so to speak, okay? And you can see it's much thicker, all right, much more robust. And then you can see this far cell in the picture here, this big blob. That's not the nucleus. That's actually the vacuole. We'll talk about that. And then that little purple small blob, that's, I think, what they're trying to show as a nucleus. And then here you can see these channels or tunnels, if you want to think of them as channels or tunnels. So these are open channels. We'll talk about them. And these are what forms between neighboring cells to just allow for nutrients and chemicals to flow back and forth. So neighboring cells can talk to each other chemically and they can share things chemically through these small tunnels or channels, all right? And we'll talk more about those. Okay, so let's go back and give a little bit more information now about the primary cell wall. So you can see here it's the outermost cell wall and it's thinner than the secondary. We tend to abbreviate it as a 1 with a superscript degree signal, symbol. And so that means primary. So primary cell walls, when you look at them chemically, what are they made of? They do have pectin, but they also have cellulose, which is the most important thing that make up the structural support of the primary cell wall. And then there's a related chemical called hemicellulose, which I'm not going to get into. I'm going to focus in on cellulose, but there is some hemicellulose, which is structurally and chemically similar, but the cellulose in terms of percent wins on that. And then there's some proteins in there as well. All right, so when you look at a primary cell wall and you look at all the ingredients, it's really the cellulose that is doing the lion's share of adding rigidity to that cell wall and some nice structural support. So let's look at that a little bit. All right, so here is a step-by-step here of what's happening on a more big picture idea and then all the way down to the chemical nature of cellulose. So let's just look at, when you get down to the nitty-gritty of it, when you look at cellulose at its simplest chemical structural, what is it? Well, if you look at this a little bit, you can see that they're repeating units of this chemical. This is glucose, all right? So C6H12O6, these are glucose molecules that are bonded together in a very particular way into long strands. And when that happens, we say it's cellulose, all right? And then what you have to appreciate in the primary and secondary cell wall, it doesn't just stay as like this single strand of glucose molecules. What happens is you get these cellulose strands or chains, they all cluster together into what we call a microfibril. So when you look at this picture and you see all those white dots in this cross section here of the microfibril, all those white dots, that's a chain of glucose, okay, long strands of or chains of glucose. So that's called a microfibril. So you have lots and lots of glucose chains that make up a microfibril and lots of microfibrils that make up a fibril. So you see that you have this pretty robust, intricate design of these cellulose molecules in these microfibrils and fibrils. It's really ingenious because now in that format, in that three-dimensional manner of organization, makes the primary and the secondary cell wall super strong. Really great design, all right? So this idea of chains of cellulose that make up the microfibril all clustered together, that's a microfibril. Then you cluster all the microfibrils in, that becomes a fibril, all right? And that's what you have wrapping around the primary cell wall, or the cell wall. You have the primary cell wall wrapping around the cell membrane, sorry. All right, so really neat design. Really accomplishes the goal of nice structural support, okay? So cellulose and hemicellulose, in this picture, they're showing it, you know, as these kind of brownish red structures, and then the hemicellulose is kind of laid down around the cellulose, and in this picture, it's a blue. But then you see that there's also proteins involved and pectins, all right? And so the way this is designed, the way they drew this, I like to use the analogy of if you've ever been to a construction site and you see someone, the workers are pouring reinforced concrete to maybe make a wall or the basement floor or whatever. Reinforced concrete is very similar to this idea where the cement is, you know, when the cement mixer comes to the site and it's got that big thing on the back of the truck rolling and the liquid cement in there, and they have to keep it rolling to keep it, you know, liquid, and then they have the rebar, I think it's rebar, right? Those steel bars, they call it rebar, that are laid down to make the basement floor, you know, design or the wall design, and then they pour the concrete or the cement, I'm sorry, the cement into that and let it harden, and then it becomes what we call reinforced concrete. So if you think of that idea, that's very similar to what the cell walls kind of anatomy is in relationship to reinforced concrete, where you have the cellulose are the rebars, the steel, and then the pectin and the proteins are the liquid, you know, cement, so to speak, that will kind of fill in everywhere and then harden up. Okay, secondary cell walls, we still see some of the same major chemical players. There's pectin in there, but typically the percent value is lower. And the extra component that you do not see in a primary cell wall is lignin. Okay, so lignin only shows up in secondary cell walls, and there's a reason for that. It is a highly complex chemical molecule. It's a polymer. They didn't figure out lignin structurally, chemically for ages. It took forever because it's so complex. It's a branched, highly branched molecule and made of sugars, and it really adds extra rigidity. And so the secondary cell wall is even more rigid and structurally sound than the primary cell wall. Okay, so very, very dense compared to the primary cell wall. And it, like I said, it's laid down or made to the inside of the primary cell wall when you look at the plant cell in total. And secondary cell walls, so when a cell is, like I said earlier, the cell finally reaches its maturity, it's, it's fully formed inside, it's got, the necessary proteins and enzymes and internal structures made, it's got its full physical size, this is as big as I'm going to get, I'm done growing. And then gene expression on some of the cells will shift and they'll say start making a secondary cell wall. And they have to save that for last, because what happens is after the secondary cell wall gets laid down, the cell then gets the signal to die, all right? And that's not a bad thing, all right? It just means that that particular cell, the internal components of the cell will die off and you're left with kind of like a hollow husk of a cell, but it's very rigid there. You still have the cell wall. And so it's an area of the plant that might not have an active cell there, but it's got that structural rigidity in that section of the plant body. So it's a very nice way to lend strength to certain parts of the plant body. All right, so the plant dies after the secondary cell wall is fully formed. But it's for a reason. It's not just dying because it's sick or something like that. It's for a specific reason that it's doing that. And I'll get into that. I'll point out what parts of the plant body are, these cells are dead, but it's a good thing. It's supposed to be that way in order to help the plant be optimally designed for its habitat.
So is lignin found in all like vascular plants or all plants or?
Any plant that has a secondary cell wall, so pretty much any plant yeah, lignin's there.
Does that include like non-vascular plants?
So non-vascular, when we use the term non-vascular, the non-vascular idea is that they don't have xylem or phloem, the vascular tissue, the blood, like the blood vessel system.
Yeah.
And so actually, when we get into xylem tissue, xylem has a ton of lignin in it. However, some non-vascular plants do have secondary cell walls in other parts of their body.
Okay.
So, yeah, there's still lignin there, just not nearly as much as what you would see in a vascular plant.
Okay, and then I know the word lignin comes from like the word wood, and I'm assuming at least because it sounds like the word wood in Italian. Is there like a lot of lignin in like wood then? Okay. Is that, or like bark? Is that why it's really hard then?
Absolutely. Okay. In fact, I was gonna say that, when they make paper, so the Upper Peninsula actually, I don't know how much currently this is happening, but when I was in grad school, I went to Michigan Tech for my PhD. And I actually worked with red pine trees, and back then they were called the Mead Paper Company. They're called something else now, but they had several, there's something else, I can't remember what they're called, but they had red pine plantations through the whole UP to grow red pine trees for paper production. And so they would take the tree trunks and send them to the mill. Of course, there's a lot of lignin, in the tree trunks, and that created a problem because lignin is so chemically stable and complex that we need to make paper from a tree trunk. That's why, have you ever driven by a paper making factory? It smells bad. You drive by and what is that smell? It's because they're using some really nasty chemicals to break down the lignin and try to get rid of a lot of the lignin. So because if you have paper that turns yellow really easily over time and it's really brittle, that has a lot of lignin in it. And then like there's different grades of paper, like you can get well, I don't remember what they call them, but there's different grades of paper, and it basically means the higher the price, the less the lignin in it. And then you can get the stuff that doesn't have any lignin in it for things that you want to preserve like forever, like your graduate thesis or something like that. So yeah, wood has a lot of lignin, and that can be a problem when you try to make paper from it.
Thank you.
Yeah. So, all right, let's talk about communication between neighbors here. So now we're looking at 4 cells that are neighboring each other, and they're showing in this picture these green little tunnels and channels between the cells, okay? So what do we got going on here? The actual channel or tunnel that connects 2 cells is called, well, in plural, the plural is plasmodesmata. Plasmodesma is singular. If you just talk about one channel, we rarely do that, so we talk in plural. So plasmodesmata, I do need you to know that term as well. All right, that's going to come up through the whole class. So the plasmodesmata are the tunnels or channels that connect neighboring cells. And what they're showing here is that this green stuff that you see that is kind of flowing from one cell to the next, that's just essentially membrane tissue that basically these tunnels have membrane tissue that line the tunnel but never cross. They never do a cross barrier. So this is an open channel. There's no membrane that these chemicals have to flow through, you know, and pass through. They just flow through the tunnel that's lined with this membrane material. Okay, so it's an open channel. It's important to recognize it's an open channel. And so there's really no like way, there's no guards there to say you can't come through and you can come through. It's just a physical size thing. So whatever's small enough to fit through can fit through. All right, there's no gated system here in any way. All right, so we have the plasmodesmata, really, really important. And then the other thing you're seeing here is the vacuole is showing up in all of these plant cells. And the artist drew it correctly. These are probably mature plant cells. When you think about a plant cell that's reached its full maturity, it's not the nucleus that's the biggest organelle inside the plant cell. It's the vacuole. In fact, the vacuole is massive in compared to all other organelles. And in plant kingdom, when we would look at the plant tissue under the microscope, often you would see, like you can see the chloroplasts typically, because there's usually lots of them in the leaf tissue, and you see the chloroplasts all like squished off to the side of a well-hydrated plant cell, and you're like, oh geez, sorry, my dog is yelling at my cat. Why are, you know, the chloroplasts all smushed off? It's because the vacuole, oh come on, hey, sorry, the chloroplasts are smushed off because the vacuole is so full of water that it's swelled up and it's pushing against everything. All right, so the vacuole is another tool that the plant has in order to create some rigidity to the plant body. So I'm sure you've gone to a store and bought, you know, lettuce or arugula or kale or whatever, bring it home, and then you take it out of your refrigerator and it's all wilty. Well, now the vacuoles have very little water in them, and so it caused the tissue to wilt. But if you slap that into a bowl of water and let it sit for a while, hopefully the plant tissue will suck up some of the water, and then the vacuoles will plump up. They'll start pushing against all the components inside the cell, which then push against the cell membrane, which then push against the cell wall. And so you get this nice, plump, rigid plant tissue. All right. Same with if you forget to water your plants and they're all droopy. You can imagine then when you do water the soil, eventually that water gets into the vacuoles and really plumps up the leaves and lets the leaves finally get more upright and, you know, try to orient their leaves so that they're facing the sun better. And so that vacuole is really, really important for structural you know, rigidity, to give that rigidity to, especially to leaf tissue, but it also stores things besides just water. Okay, there's water in there, that's the main ingredient, but there's other things like pigments. Certain colorful pigments tend to hang out in vacuoles, but pigments can hang out in different parts of the plant body, but vacuole for the water-soluble pigments, there's enzymes in there and proteins. If you've dropped an apple recently, I hate it when you drop an apple and then like minutes later you get a soft spot wherever the apple, you know, hit a sharp edge or something. Well, that soft spot on the apple, what you did there is you took, like if this was the apple tissue, what you did when you dropped that apple is you busted open all these vacuoles because of the, you know, the harsh physical impact. Well, there's enzymes in these vacuoles and they start to break down the plant cell tissue. And so you get that soft spot. All right, so vacuoles store up a lot of stuff, and vacuoles can also store up things that might be poisonous to the plant. And it's a way for the plant to sequester things that aren't so good for them and keep it away from the more precious proteins and enzymes that are floating around in the cell. Okay. This picture down here is just showing an electron microscope picture of the plasmodesmata that are connecting to neighboring cells. Okay. So really, really important that they have a way to communicate to neighboring cells and allow for chemical flow, water flow. Another picture showing the plasmodesmata between two neighboring cells. And again, just trying to show that there is a membrane that lines the tunnel, but there's no cross membrane. So it's just a free open tunnel. They can just have the chemicals sailing right through it. Is there a question?
Yeah, the middle lamella, is that like between the primary and secondary cell walls or between the cells or both?
Between the cells. So, the primary cell walls is the most outermost portion of the cell.
Mhm.
So I guess technically it would be between the two primary cell walls of neighboring cells.
Okay,
Okay.
thank you.
Yep. Yeah, if anyone else has questions, I can't, like in my view right now, because I'm looking at my PowerPoints, I only see a handful of students. So if you have a question, you can unmute and just, interrupt. I'm fine with that.
I have a question.
Yeah.
So I'm just curious, is this bad for cells when it comes to them, or plants when it comes to them being sick? Like, is this just a free path for the virus to roll through there?
Ooh, good question. And the short answer is yes, unfortunately. You can get viruses, certainly can fit through the plasmodesmata, you bet. And so once a virus gets in, and often a virus can get into the plant through an insect that's feeding, often you think about aphids and things like that, that have that stylus mouthpiece that's like a needle, essentially, and they can pierce right into certain parts of they often like to go into the vascular tissue to suck away the sugars. And so viruses then get through the saliva of the insect into the plant, and then the virus can go through the whole vascular tissue. It's like an internal highway system that now they have full access to the plant. Plus, they're small enough to fit through plasmodesmata. So yes, you can see that pathogens, that is a way for if they're small enough to fit through the plasmodesmata area, they can certainly use that as a route to get to new territory. Unfortunately.
Aphids are the mosquitoes of the plant world.
Aphids, yeah. I tend to, I grew up around, the wild. I lived in the UP my whole life, so I am a forest girl and lived out in the woods a lot at the camp. And so I kind of grew up with the live and let live, but there's certain insects now, it's like, kill, kill. And aphids are one of them because they can, in the greenhouse, especially all of our greenhouse plants, when you have all these plants in a enclosed area, it's very easy for insects to get out of control and spread. And so those, I'm okay killing those.
So is the plasmodesmata something that like a mycorrhizal relationship is going to take advantage of?
So typically not. So there's two main categories of mycorrhizal fungi, and the one type that's called the ectomycorrhizal fungi would not at all interact with plasmodesmata. Now I'm just thinking about the other type, the endomycorrhizae. I guess well, I don't know if I'd have to check their diameter, the fungal diameter, and compare it to the plasmodesmata. That's a good question. It's really, I'm trying to debate if the hyphal diameter is small enough.
Don't they go around cells, not through them?
Right. They never physically, so very good. Someone must have been taking Plant Kingdom last semester, because you'll learn about this again in this class too, because I make a big point that endomycorrhizal fungi, they're a little bit more intimate with the plant, but they never break into the plant cells where they're like floating around, like, we float around in a swimming pool, where they're just free and they're interacting with the cytoplasm. The fungal hyphae never do that. And so they're always surrounded by a plant membrane tissue, or by the plant membrane of some sort. Usually that's the cell membrane that totally wraps around the fungal hyphae, so that the fungus can be kind of protruding into the cell area, but it's always encased in a membrane designed by the plant. And that is so that the plant can stay in a good relationship with the fungus. If the fungus were to breach that membrane and get in, free swimming into the cytoplasm, that's a signal to the plant, oh, that's a pathogen. It's totally invaded, it breached my barrier, this is too intimate, it's after all the goodies I have in my cytoplasm, so I'm going to start fighting it and send up my defenses. All right, so we'll talk about that. It's really an amazing interaction between the fungus and the plant. It's really a dance they have to play in terms of communication, and then the structural shifts that take place, both with the fungus and the plant in order to accommodate each other. It's really cool. So we'll get into mycorrhizal fungi. We do talk about them in this class. I put this here. It's a design, again, to show, here's a plasmodesma. I guess they're only showing one. So plasmodesma here between two cells. But I wanted to make the point here, in this drawing, whoever drew this, they had in their mind a baby cell, a really young, immature cell, because you see the plant vacuole is still really small. All right, so I just wanted to point out, you'll see this in textbooks a lot. And now that you know more about plants, I want you to be aware that when you look at a mature plant cell, this is not a correct drawing. A mature plant cell, this vacuole would be massive in comparison to everything else. All right, so whoever drew this was thinking about a baby cell that hadn't quite reached its full maturity. All right, so just be aware of that. Now this is more, whoever drew this, whatever artist did this one, they were thinking of a mature plant cell, because here you can see the vacuole is huge. You see that it tends to dominate the central portion of the cell. And this is what I mean by things being squished off to the side. All the other organelles tend to get squished off. But this is good if that, and they're showing a nice plump cell. So this vacuole is full of water and it's pushing against the cytoplasm and all the organelles. And so all of that's getting smushed up against the cell membrane, which is getting, pushed against the cell wall. So you get that pressure. It's called turgor pressure. So when we talk about pressure because of water buildup, that's turgor pressure. And that's really important for especially the plant leaves, to stay upright and to be able to turn and face the sun and get access to the sun. So having well-hydrated plants helps them do photosynthesis, because it allows them to orient their leaves correctly and make sure that they're facing the sun for optimal photosynthesis. All right. Another drawing with a little bit more detail perhaps. And again, they're trying to show that the vacuole is the largest component of the cell. And there's others, certainly we're going to talk a lot about chloroplasts in this class. They have mitochondria too, just similar to our mitochondria, because they have to carry out respiration just like we carry out respiration. And unfortunately, we don't have chloroplasts. Wouldn't it be awesome to have our own chloroplasts? We wouldn't have to eat. Just go to the beach, soak up some sun, and we're good to go. And then here's the idea of water in or water out. What happens to the vacuole? So if you have nice plump lettuce leaves that you're eating for your salad, you can imagine next time you bite into that the vacuoles are full of water, nice and plump, pushing, that turgor pressure is pushing against the cells, and it tastes good, feels good when you're chewing it. However, if you let your lettuce stay too long in the fridge and dehydrate, this is what's happening. The vacuole starts losing water. You get that gross limp lettuce that's not good texture either when you eat that. So we'll talk a lot about water movement in the plant and how that's happening. It has a lot to do with osmosis. So if it's been a while since you've thought about osmosis, you might want to brush up on that, because we're going to get into it actually on Monday. So we're going to get into that. All right, chloroplasts, like I said, we're going to get way into that in this class. When you look at a chloroplast, this artist drew it correctly, there's actually two membranes. There's this outer membrane, there's this inner membrane, that's in this sketch, it's orange. And then there's a third membrane that's internal, all right? So this internal membrane that's all green, that's called the thylakoid membrane, okay? Thylakoid membrane, I think I have that on a subsequent slide. We're gonna get way into that, all right? We're gonna be talking a lot about the chloroplast and the thylakoid membrane, and then also the watery portion that is surrounding all of that thylakoid membrane that the thylakoid's, floating around in. So we're gonna get pretty intimate with the chloroplast and all of its anatomy. Really, really important structure. So there's the spelling of thylakoid. It's a weird spelling, of course, like pretty much anything in science. So that term I definitely need you to know. So let me just wrap this up here. So the thylakoid membrane, there's essentially two different parts to it when you look at the drawing and how they use their shape. So you have a relatively unstacked region that, to me, kind of like a bridge between two stacked regions. So you have these relatively unstacked regions like a bridge. That is called the stroma lamella. All right, I do need you to know that. And then there's stacked regions. Okay, the stacked regions are called grana. Plural is grana, singular is granum. All right, so the stacked regions of the thylakoid are the grana. The unstacked regions of the thylakoid are the stroma lamella. Taken all together, if I just point at that whole internal membrane and I say, what is that internal membrane? You would call it the thylakoid. All right, if you talk about it in total, it's the thylakoid. And then each part of the thylakoid has a special, stroma lamellae portion, the grana portion, and then all of that, the thylakoid, that's an internal membrane. It's distinct, separate membrane from the outer and inner membrane. It's its own membrane inside, and it's floating around into the watery matrix that we call the stroma. All right, so the stroma is the watery portion. If you cut open the thylakoid membrane and peer inside of it, it's hollow in there, kind of like our intestinal tract. That is called the lumen, and that's what we call the inside of our intestinal tract. We say it's the lumen portion of the intestinal tract. So that hollow internal portion of the thylakoid, when you peer inside of it, we call it the lumen. You do need to know these terms because when we get into photosynthesis, I refer to things that are happening in the lumen, things that are happening in the stroma, things that are happening in the thylakoid. Okay, so start getting familiar with that now, please. It won't go away. All right, I think we've run out of time. So plant anatomy, some very different things compared to animals, but when you look at it, very well designed for their habitats and for their purposes. So we'll stop there for today. Please look for an email from me over the weekend announcing what's going to happen on Monday in terms of who's showing up for lecture, and we'll do a synchronous Zoom with the people who cannot be present. All right, so I'll see all of you Monday at noon, whether it's face-to-face or via Zoom, okay? So have a good weekend. Stay safe, everybody. Be careful.
Thanks.
Thank you.
Bye-bye.
Thank you. Have a good weekend.
Bye.
You too.